And we would like to thank you for attending today's webinar, Hope for DCM Therapies, Tenaya Therapeutics. We really appreciate each of you taking time out of your busy schedule to be with us today. I'm Greg Ruff, the board president of the DCM Foundation, and our presenter today is Dr. Whit Tingley, Chief Medical Officer at Tenaya Therapeutics. Whit's experience includes Vice President of Clinical Development at Cytokinetics. Whit, did I say that correctly?
Yeah. Perfect.
In his time at Genentech, he was the therapeutic area lead for cardiovascular and metabolism research and early development. His experience also includes medical and group medical director roles at Genentech and Roche, and he was an adjunct associate professor at the University of California, San Francisco. And he is a really, really nice guy, and I'm glad that we have him on our team in our fight against DCM. Both organizations are aligned with really the thought of giving hope to DCM patients and family members. So we're very excited to be here today with Whit and the folks at Tenaya. We'd also, the DCM Foundation and its board and all its members would like to thank Tenaya Therapeutics for their generous sponsorship to support our mission and the mission of DCM. A few housekeeping items here. We're going to have, I believe now, 6 actual audience polls throughout the next hour. We'd love if you would actually participate in each one. Very simple, very easy to do. Poll pops up, click on the question that best suits you, and then we'll tabulate the results. So please pay close attention. Also, we'd love to get any questions from you throughout today's presentation. You simply just look at the chat box at the bottom middle, basically, of your screen, click on it, type in whatever question you have, and hit the enter button, and that'll be sent just to the panelists, so it will be anonymous, but we will actually take some time out during the presentation and at the end of the presentation to address the questions that you have. We did this last month, and we wanted to repeat it again. We want to see who's here today. So we'd love to get an idea of who you are and why you're here. Are you a patient, a family member, medical professional, or interested for another reason? We'll give you about 15 seconds here. Click on what most suits you on the submit button, please. Interesting. So we've got a little over a third of the folks are patients, about 1 in 5 family members, a little under 1 in 5 medical professionals, and then other folks. So again, looks like we've got a great range of people with us today. In what today's presentation is about, I think, as I mentioned before, what the DCM Foundation and certainly what Tenaya is all about is hope for DCM patients and their family members. As a DCM patient and as a DCM patient for a number of years now, I realize that there is no cure for DCM, but given the brilliant people like Whit, his other scientists at Tenaya and other organizations like Tenaya around the world, I have hope, and I believe very strongly that in the future, in the very near future, additional advanced therapeutics will be developed for DCM patients, and at some point, even a cure. So I hope that after the presentation, we've given you some more hope about this disease. One thing I want to also add is, given the deep scientific medical nature of what Tenaya does, there's a bit of technical information on it. We don't expect you to understand everything. I certainly don't. We really wanted to show exactly the complexity in what Tenaya was doing. So don't worry if you don't exactly understand everything. If something pops up and you have a question, again, certainly enter it in the chat box and Whit can go into more detail about it later. And also, there will not be a test at the end of this presentation, but please pay close attention. I'd like to present now to you Dr. Whit Tingley. Whit?
Thank you, Greg, for having us and giving us a chance to talk about our team and what we're working on, and how we hope to bring hope to patients with DCM. But thank you even more for your work setting up this foundation. You and Jenny, who's helping run this now, are doing amazing work to reach out to patients and bring the community together and assemble all the pieces. I've dedicated my career to using science to provide new options to patients with heart disease. And I've worked in academic university setting, I've worked as a practitioner, I've worked in industry. And I know that it takes a partnership of patients, families, academic experts, and industry to bring everything together to actually get new options into clinic for patients. So this is really where it starts, and thank you for founding the foundation. So today we'll introduce our company briefly. We'll do a deep dive into the science. And as Greg says, we're going to provide a lot. I know people in the audience have different levels of scientific background, and my hope is to give something interesting for everybody. So please take it in that spirit. We're going to highlight our efforts focused on dilated cardiomyopathy and how we leverage genetics to find new therapies, both targeted for specific forms of dilated cardiomyopathy, as well as ones that might apply to other forms of heart failure as well. And then we'll conclude with how you, patients, family members, and others in the community can get involved right from the beginning. So as you know, work is hard. But hard work is great when you have a real purpose. And for us at Tenaya, you give us that purpose. We're very inspired by your courage and how you cope and handle heart disease, heart failure, and dilated cardiomyopathy. And it really helps us to hear your stories, hear about what's working for you and what's not working for you. It gives us directions to head, but also gives us the passion to continue doing what we're doing. So we look forward to building communications and collaborations going forward. So we are a small startup company with 70 employees now and growing quickly. We were founded in 2016 with the mission to focus 100% on heart disease. And we were founded by some of the most known molecular cardiologist researchers in the world, with the mission of bringing state-of-the-art science to this problem of heart disease, starting with heart failure. In addition to our founders, we have a really quite inspiring group of advisors, and even our board members and financial contributors are themselves very accomplished scientists. So our board meetings often end up going into deep scientific discussions, which is really the way we like it, because the better we do the science, the more likely we are to come up with a new option that actually helps you and other patients. So with that, I'll go into more detail about the science. We are 100% focused on heart disease, but we have multiple platforms within that. We'll talk today about gene therapies, about heart regeneration, which is growing new heart muscle, and then our precision medicine platform, which is how we aim to address specific genetic causes of dilated cardiomyopathy. With gene therapy, the main problem is to fix a broken disease, to prevent genetic disease onset and to reverse it once it's started. This is a growing field across medicine, and we have new ability to get normal genes into the cells that need them to fix broken genes. Regeneration is addressing the problem of loss of heart muscle. As you may know, many cells in the body can divide, but heart cells are not included in that. After just a few weeks of life, we have the cells that we're going to have. So after a heart attack, or in many forms of heart failure where heart muscle cells die, the body can't replace them on its own. Our founders discovered a new technique to take other cell types in the heart that do divide and replace themselves, and reprogramming them using genes to become new heart cells. So we are developing this technology to regenerate lost heart muscle. And precision medicine, which we'll do a deep dive into first in this talk, is treating the underlying cause of DCM in the heart, and specifically using genetics to tell us what those underlying causes are. And we start with cells, including cells derived from patients. There are ways in cell culture to turn those into beating heart cells in the dish. And when we look at the mutations that cause cardiomyopathy, we see that they also cause changes to the cells, abnormalities in the cells in the dish. And this gives us a platform to test many, many different chemicals to figure out which might work as new drugs. So this slide will break down a new way of talking about the three platforms we just discussed. So gene therapy, for example, is a one-time treatment. You get a single dose, and some forms of it are delivered intravenously, and some forms are infused locally in the heart with a procedure like a catheterization procedure, like getting a stent, a heart stent. The delivery vehicle is a virus. So we're all very aware of viruses these days with COVID-19. But it turns out there are other viruses that don't cause any disease that people still catch and have. And those can be harnessed and repurposed. So the machinery that lets them divide is removed. They can't survive on their own anymore. But it's replaced with normal heart genes. And the virus is the way that gene can get into the heart cells. So with that virus, we can deliver healthy copies of genes to the heart cells that have the genetic mutations, and counteract the mutations or correct the mutations. That's the basis of gene therapy. For regeneration, we are reprogramming other cells in the heart to squeeze like heart muscle cells. And to do that reprogramming, we are delivering genes. So it's actually ends up being very similar to gene therapy. We are using a similar virus to deliver genes into, in this case, cardiac fibroblasts. That's what the CF stands for. And reprogramming those fibroblasts to become cardiac myocytes. This would be a one-time treatment as well. In this case, it requires injection into the heart muscle. And then our third platform is the precision medicine platform where we are screening for new drugs using cells in culture, including cells derived from patients with DCM. And in our first iteration of this, we've been screening for new drugs that would be small molecules, which means they can be taken as pills. And we are finding what chemicals can take the abnormal cells in the dish and make them look structurally normal. And then we test in animals, and then we'll bring them to clinical trials. So that outlines our 3 platforms. Our first Tenaya poll is just your gut feel from what you've heard so far, and we will go into a deeper dive, but from what you've heard so far, which of these platforms do you think are going to be the most important for new therapies going forward for DCM in your community? Is it gene therapy, regeneration, precision medicine, all of the above, or none of the above? And as you vote, I will just remind you that gene therapy is placing the healthy copy of a gene into the heart to replace a broken copy. Regeneration is growing new heart cells, for example, after a heart attack. And the precision medicine is the guided platform for personalized medicine based on gene mutations. Okay, that's a good mix. All of the above is the winner. So obviously, we like that answer because that's what we're doing too, all of the above. And precision medicine and gene therapy are near tied in its second. Thank you. Greg, Jenny, have you seen any questions so far? If not, we'll move on to a deeper dive into the research.
We had one question come in. In gene therapy, can you fix genes before they begin to mutate or develop the disease?
So no and yes. So you're born with the mutations. Either you inherit them from your parents, or in the early development, or in the egg or sperm from your parents, there's a new mutation that you're born with and developed even in utero with. But yes, the vision for gene therapy is to prevent disease before it happens. Now, the vision for developing a gene therapy typically starts with people who are already affected or are very close to having problems with the disease. And then we establish the efficacy and the safety in those people that have the most to benefit in the nearest term. And then as we establish the safety and find the right dose and so forth, we can bring it earlier and earlier in the course of disease into more of the prevention mode. It's a great question.
We have a couple more questions coming in, but they're more related to time frame, and I think you'll address them later in the presentation, so we can wait on those.
All right, so we'll come back to those at the end. So there are really good therapies for heart failure, including DCM, life-saving therapies. As you know, you can have quite a few pills to take every day. And I strongly encourage you to follow the advice of your doctor and take your medications. The interesting thing is that all the medications we have for heart failure are targeting different things than the genes that are mutated in genetic dilated cardiomyopathy. So the pills you're taking likely include beta blockers and ACE inhibitors or angiotensin receptor blockers, aldosterone inhibitors. Those are neurohormonal antagonists, where neurohormonal is the word basically that means hormones, which are signals between cells. On the flip side, all the genetic mutations, or the vast majority of genetic mutations, and there are over 50, that we know of that cause dilated cardiomyopathy, act on proteins that are inside the cardiac myocytes, They're not secreted, they're not hormones, they're not signals, they're doing other functions inside the cardiac myocyte. They're like the machinery. Many of the mutations are in the machinery that actually does the contraction. So the drugs that we take now aren't directly affecting the underlying cause of the disease in the case of genetic cardiomyopathy. So what Tenaya is trying to do is directly address the underlying cause, starting with figuring out what the genes are actually causing, the problems they're causing inside the heart cells, and then finding ways to prevent or reverse those problems. So as I said, there are over 50 different genes known to cause DCM. And this is a schematic highlighting many of those genes. Each DCM gene has a yellow star, and it has a name like PLN or BAG3 or LMNA. These have different functions. So this is a picture of the sarcomere, which is the machine that does the pulling inside the muscle. And many of the mutations are in the sarcomere. There are mutations in things that pump calcium around the cells. So PLN we'll talk about more, and SERCA2 is involved in this. There are genes that are involved in protein folding and shaping. And we'll talk about BAG3 more in this talk. And then genes involved in holding the cells together. Interestingly, mutations in these genes can cause arrhythmias as well as heart failure. And then genes in the nucleus as well, including LMNA. So we are starting with some of the more prevalent causes. This table shows an estimation of the frequency of DCM caused by each one of these genes, with Titin or TTN being the most common. So we're starting with a subset, but our ultimate vision is to develop different treatments tailored to the different gene mutations. And in a few years here, we hope we'll have some pills for certain types of gene mutations, like gene A, B, and C, generic names, or gene therapies for other genetic causes of cardiomyopathy, and then potentially other avenues like biologics as well. So this is one more reason why it's so important to have genetic testing. It helps all of us understand where the needs are, how common the mutations are, and get started on figuring out what those mutations are doing wrong. So our discovery platform is based in part on cells that have been donated by DCM patients. There's a new technology that actually won the Nobel Prize a few years back to make stem cells from regular cells, including blood cells. So DCM patients who contribute to blood samples, those cells from that blood can be turned into the specialized stem cell, which is called an iPSC, for induced pluripotent stem cell. And what we can do with those cells in the dish is differentiate them so that they become heart cells or cardiomyocytes. So that's schematized here. And what we end up with is iPSC-CMs, or induced pluripotent stem cell derived cardiac myocytes. And we can study these in the dish. And Tenaya has over 20 cell lines to date from actual patients with severe cardiomyopathy who unfortunately had to undergo transplantation before they were 30 years of age. So these cells appear abnormal in culture because of their mutations. This may not project as well as I had hoped, but these are cells. Each blue dot is a nucleus, and the green staining is the contractile apparatus, the sarcomere, the proteins that are doing the contraction. And what you hope to see at this low magnification is sort of a uniform green haze. So that's because the very fine distribution of the sarcomeric proteins. In this example, in cells that are mimicking patients with BAG3 mutations, you can see it's chunkier, right? It's there are bright spots and dark spots due to more aggregation of the proteins. And then there are breaks in the filaments which are the structures that do the pulling. So what we can do in the dish is add chemicals, small molecules that are potential drugs, and we can add thousands of chemicals and look for the one, and this schematized here, the one chemical that improved the appearance of the cells. You can see it's gone back to the more normal appearance of the sarcomere after the treatment with this drug. And we can quantify this effect using machine learning artificial intelligence, which allows us to screen these thousands of compounds. So this experiment actually gave us our first lead potential drug. And we've brought that drug and treated mice to see if it improves heart function in mice. So this same mutation we talked about with the cells in the gene BAG3 also causes heart failure in mice, just like in humans. So here, many of you probably know ejection fraction and may know your own ejection fraction. It's one of the best measures we have for heart function. And a mouse, normal ejection fraction is around 70–75%. When mice have mutations in the BAG3 gene, their ejection fraction is low and continues to fall as they get older, and they eventually die of heart failure. Treatment with this potential drug that we discovered on the last slide stops the progression of the disease in mice. So at this point in experiments, we've seen improvements by more than 20% in ejection fraction. So that's as if your ejection fraction went from 35% to 55%, which is, as many of you may know, a big deal. Indeed, this compound improves survival in mice, and no safety concerns have been identified to date. The mice tolerate it well. There's clearly a lot more work to be done for safety, but promising so far. It actually works very differently than the pills that you're taking now. We are testing it in combination with current therapies. We know that the targets are different. The actual protein that this chemical binds is different than the hormones and hormone receptors that your medicines bind. One advantage of this particular target is that we can use blood testing to determine whether we are hitting the target as expected, both in animals and ultimately in clinical trials as well. And we've discovered this in BAG3, as I've described, but the reason we use BAG3 is because it's involved in protein folding, which is a problem in many forms of genetic and non-genetic heart failure. So we will go on to test this particular potential drug in types of heart failure beyond those caused by BAG3 mutations. So we are encouraged by the results we have so far. We still have a long way to go with efficacy and safety studies in animals, and then starting in clinical trials. Now, we've talked about the cells derived from patients, and they give us our first opportunity to test whether this particular potential drug works in other genetic forms of heart failure. As I mentioned, because it works in a way related to protein folding, we expect it will help in other genetic abnormalities where protein folding could become the problem. So we're testing it in the context of titin mutations, actinin mutations, and LMNA mutations, for example. And we have a variety of scoring systems and different metrics from these cells to assess whether it's working. But the other thing our panel of cells provides is a way to discover new drugs. So this potential drug was discovered in BAG3, but we will use titin and LMNA and other cells to do whole new screens to identify new potential targets. We are very fortunate to be collaborating with the DCM Foundation and also with the DCM Research Project, also called the DCM Consortium, which was founded and still run by Dr. Hirschberger. Patients participating in this project donate blood samples, and Dr. Hirschberger and his colleagues around the country store those samples and have shared them with Tenaya. We have successfully made iPSCs, the induced pluripotent stem cells, from these samples and turned them into heart cells in the dish. So this is a really valuable resource, not just for Tenaya, but for both academic research and the biotechnology industry together. So just a big shout out to all the patients who've directly participated, as well as the families and other loved ones, and scientists, of course, who are contributing to the DCM research and the DCM consortium in particular. So poll number 2, have you ever participated in any of the following types of research? And you can hopefully check more than one. So the dilated cardiomyopathy research project that we just discussed, other heart failure research studies, other types of clinical research, all of the above or none of the above. All right, good participation in the DCM research project. That is great to see. Thank you, everyone. And other types of research. It's interesting, the heart failure research is lower. There are actually quite a few pharmaceutical companies working on heart failure research now, and there's ample opportunity to participate in different types of trials, both registry or natural history studies, where there's not an experimental treatment, but we're learning more about the disease, and in interventional trials where a potential new treatment is being tested. So thank you for everyone participating, and I would encourage everyone to continue to participate. We're going to switch now to gene therapies, but it's a good opportunity to ask if questions specific to this section have come up.
We had 2 questions come in that I think could be addressed here. First one is, can the solutions that you are working on help not only the muscle contraction area, but also the arrhythmia issues that DCM patients might experience?
That is a great question. Yes, but it's a little complicated. Abnormal heart rhythms, in the worst case, sudden cardiac death is a huge problem, and particularly in dilated cardiomyopathy. And there are forms and particular genes that are even more prone to this, and syndromes called arrhythmogenic cardiomyopathy. So when we are approaching the underlying cause of the disease and looking at these genes and how they're working in cells and how we can correct that, we hope and expect that will correct the underlying problem that leads to both the heart failure, the reduced contractility, and the arrhythmia. Now, one caveat is that once there's fibrosis in the heart, once the cardiomyopathy has set in, that risk of arrhythmia from that may persist. So as I talked earlier about starting with patients that are already quite affected, and then during development expanding to patients in more preventative mode, it's probably the case that it will take a little bit longer till we get to the preventative mode that will prevent abnormal heart rhythms. But we are focusing on genes that cause arrhythmic cardiomyopathy, as well as DCM, and genes that cause both.
And then one other question, interesting question. Does looking normal mean the same thing as working normally?
Yes. So in order to do the high throughput, in order to test thousands of compounds, we needed still shots, essentially, just freeze frame pictures, trying to do machine learning on movies of the contractility it just takes a lot more computing power, for example. But there's actually, it turns out, a lot more information just in the patterns of the cells than is in the contraction itself. But fundamentally, that structure that we get in the picture of the contractile apparatus, the sarcomere, does translate into contractility. But really the best way to measure the contractility is in living beings. And so I showed you the impact on the ejection fraction for the potential drug that we discussed.
Thanks. I think the rest of the questions probably make more sense at the end of the presentation.
Perfect. So gene therapy actually has been studied for decades now, but in the last 5 years or so has really started to work. And now we've seen approvals of drugs for gene therapies, both delivery of genes to cells outside the body, then the cells are put in the body, and gene therapies where the gene therapy vector is infused in the body, for example, just with an IV treatment. As his background, many of you will know that every cell in the body has genes in the nucleus, and that mutations in these genes cause an abnormal protein which can lead to disease. And those abnormalities at the DNA level can be deletions or substitutions or duplications. This then is translated into the protein. So there's a defect in the protein sequence itself that leads to disease. The major progress in the last 5 years has been how to deliver normal genes to the cells that need them. For example, the cardiac myocytes, the muscle cells of the heart. And to do this, there's been new discovery of viruses that don't cause disease in humans, but do infect humans. So the virus's DNA can be removed. That makes the virus inert. It can't divide or grow or cause any problems of its own. And then the human gene, the corrected human gene, is put into this virus, which then infects the target cell of interest here, think muscle cell. And that gene is expressed and makes a normal copy of the protein. So it can replace a protein that's missing because of the mutation, or it could counteract a protein that's misbehaving because of its mutation. So one project we are working on for gene therapy is delivering a gene called dwarf. And this is a flavor where the wild type dwarf gene may counteract the problems caused by mutations in other genes. And this shows a study in mice that have heart failure due to mutations in the PLN gene. PLN stands for phospholamban. And this actually is an example of a mutation that causes a high amount of arrhythmias as well as heart failure. So here is a cross-section of a normal mouse heart. This is the left ventricle, good thick muscle walls, and a normal cavity side. So this is where the blood goes in and gets squeezed out. When mice have a mutation in PLN, in this case, it's an actual mutation that human patients have as well, you can see that the heart is abnormal. It's dilating and the walls are thinner. I'm not showing it here, but the ejection fraction and contractility are reduced as well. So when an extra copy of the dwarf gene is added in this mouse model, you can see that the appearance of the heart is much closer to normal. So again, the thickness of the walls, the pink walls here, is better, and the chamber size is more normal. Now, when we zoom in on the heart wall, we can see normal heart muscle cells, very pink here from the normal mouse heart. In the failing mouse hearts due to the phospholamban mutations, we can see extensive fibrosis. So this blue staining here is scarring, often caused when a muscle cell dies. The fibroblasts come in and replace it with scar tissue. So extensive scar tissue in this model, which is also true in patients as well. And again, when we add an extra copy of a normal dwarf gene, that improves the appearance and substantially decreases the fibrosis. Similarly, the dwarf gene improves survival in the mice. So this just shows over time and days that we are well within the normal mouse lifespan in this experiment. The phospholamban mutation leads to premature mortality in the mice, 100% of the mice early on. Overexpression of the dwarf gene improves survival. As you can see, we have more work to do. We haven't normalized survival, but we're headed in the right direction with this particular target. Now, phospholamban is one cause of dilated cardiomyopathy, but we're interested to see whether dwarf might work in other causes as well. The most commonly studied form of DCM in mice for research purposes are mice that have loss of the MLP gene. Similarly, as we saw before with phospholamban, when the MLP gene is dysfunctional or missing, we can see dilatation of the left ventricle and thinning of the walls. You can see it just in a picture of the whole heart. And then again, dwarf expression normalizes the appearance of the heart. And here I'm showing contractility as well. So these images are pictures of the heart over time. So the black is the blood inside the ventricle. The gray is the walls of the ventricle, and each peak is a contraction. So contraction, relaxation, contraction, relaxation. So this shows the good sharp peaks. The ejection fraction here is 75%. With the MLP mutation, mice have severe cardiomyopathy. You don't see the peaks and valleys. The heart is barely contracting. And then with expression of extra dwarf, we can see the contractions are restored. So in two different mouse models, this expression of this gene is improving function, improving the histology, reducing fibrosis, and improving survival. So we are in the process of converting this into a gene therapy that can be delivered with the viruses that we discussed. So poll number 3, if you were to participate in a clinical study of a potential new treatment for DCM, would you prefer it to be a small molecule, which would be like a daily pill, or a gene therapy, which would be a one-time treatment, potentially just an IV infusion? Okay, gene therapy has the edge. The disappointing thing is that there's not a single person happy with your current therapy. So gives us work to do. Gives us work to do.
We had one question that you might want to address now. Somebody said, maybe I missed it, but what does DWARF stand for?
Yeah, dwarf is a long acronym, but what it is a newly discovered gene. It actually wasn't known until a couple of years ago, but it makes a protein that is involved in calcium signaling. So calcium pumping in the heart is crucial for both normal contraction and normal rhythms, and it's a system that drug developers have been working to fix in heart failure patients for decades now. And it has really not been amenable to small molecule treatments or to like antibodies, biologic treatments. But this new discovery of dwarf, which regulates a calcium pump, gives real potential for a gene therapy approach to normalize calcium signaling. And calcium signaling is abnormal in genetic DCM, in non-genetic DCM, in heart failure after a heart attack, and other forms of heart failure as well. It's sort of a fundamental common problem. So this therapy has potential use in multiple types of patients with heart failure.
Great, thanks.
Okay, how the community can get involved. This shows, slide just shows Tenaya's values. And we have many values: scientific excellence, dedication, tenacity, community. But patients are first. We try to think about patients and patient needs and providing better options for patients for all the key decisions we make. You know, I'm very fortunate to work in a field where if you do the right thing for patients, you're likely to have a good business case as well, and get all the financial outcomes that are needed to continue to help patients. So we are starting now today, reaching out to you, and it's the beginning of a process of involving people who are affected by heart disease and their family members and loved ones in the conversation from the beginning, from early development. So how can you get involved? We've emphasized the real value that we and others get from patients participating in natural history studies like the DCM research project. And in particular, the samples that are contributed for research, including the cells that we use to make the iPSC-CMs. Helping us understand your journey more is very helpful. Clearly the impact of the disease, and again, sort of disappointing to see nobody answering that they're satisfied with their current therapies. I'm hearing that loud and clear. Diagnostic challenges also, in order to be helped by new options, patients need to get the correct diagnosis, and in genetic diseases, that often isn't happening enough. So understanding the challenges that you've had being diagnosed will help us find more patients who we can offer options to. During clinical development, there's a big role for patients to play, helping us communicate about the safety and potential efficacy of trials, of drugs in trials, and also communicate that the trials exist. But this way that patients and families can have informed decisions, not just what the sponsor, like Tenaya, it thinks about a potential new drug, but what actual patients think about it as well. Reviewing the clinical study design and making sure that we're measuring the things that are most important to you when we study our potential new drugs. And then talking with regulators like the FDA about the impact of therapy and how the results of a study show that a new treatment may or may not help you and meet one of your needs. And then after approval, so in phase 4 of clinical development, helping companies understand barriers to access, why patients may not be getting this option, and then participating in long-term follow-up to see if there are other types of patients that might benefit and to further establish the safety profile of a new drug. So this picture shows Greg when he came to our offices in South San Francisco. Our team was very inspired, both by Greg's personal story, by his family's story, because his family has been very impacted by this disease, and by his vision for the DCM Foundation. But we welcome, when COVID is over, we would love to host more of you to come and share your stories and inspire us the way Greg did. Other ways are to participate in focus groups virtually. We really regret that the foundation wasn't able to have its in-person meeting. We had intended to be there talking with you in person. And we will do that in person when we can, but virtually until then. And then telling us, what you need. If there are activities that we might be able to help sponsor, let Greg and the foundation know. And the foundation may contribute, and Tenaya as well, or let us know directly, and our patient advocacy email is shown there at the bottom. So that concludes the Tenaya section. Jenny, do you want to do questions now, or do you want to go through the last couple of slides here?
I think we can go through the slides and do them at the end.
Perfect. Greg?
Thank you so much, Whit. That was great and very inspiring, and you know, I think everybody here has learned a lot, and we really appreciate you and everyone's work at Tenaya on this. We'll run through this, and I know we got some questions we want to get to. Another quick poll. We're going to be doing 9 to 12 webinars each year. So for our 2021 webinars, we'd love to hear what you would be interested in covering. And so Jenny, throw the poll up. You can select more than one of these, select as many as you'd like. And we'd love to see what you'd be interested in so we can accommodate you and create webinars around the topics you're interested in. Great. Looks like it's pretty diverse, and clearly there's a few that have 65–70%. So we will look at this, take it into account, and get back with you very soon on our 2021 webinar lineup. Next slide, please. So the next two webinars are already in the books. The next one we will be doing in partnership with a very large sort of heart group called Mended Hearts. That'll be Tuesday, December the 8th at 7 p.m. Eastern Standard Time. And it's about staying in care with cardiomyopathy. Really excited about that. Then our first 2021 monthly webinar will be about medical devices used in the treatment of DCM. Our presenter and panelist will be Doug Raychak, which is a great expert and a patient with a device, but is very inspiring and is extremely knowledgeable in this area. Next slide. Also, I think in WIT had put up their patient advocacy. If you want to get in contact with Tenaya, you can certainly email them at the address below, and also go to their website. With the foundation, it's the same. Email us at info@DCM Foundation, or visit our website. One of the questions that was asked, Jenny, I'll get to, is will this presentation be sent? So the presentation will be sent in the next few days as a follow-up, and also a recording of the entire presentation. So if you're attending, you'll get those, and you can review them again, or certainly share them with folks in industry or other patients and family members. Next slide. Again, getting to know the folks at Tenaya, and it really has inspired me, really has given me hope. I'd love to hear, last poll question, please select one of the options below to fill in the blank. Following statement, what you've seen today with Tenaya and the Invitae presentation we had last month, does it make you very hopeful, hopeful, moderately hopeful, somewhat hopeful, or not hopeful for the future of DCM treatments? That's great. That's wonderful. Certainly, almost everyone is either very hopeful or hopeful. And again, we hope to continue to bring you information like this that'll provide hope for the future for DCM patients. Next slide, please. So Q&A, Jenny, I know you've got 2 or 3. I'm thinking if we go over a few minutes over, if you want to jump off, that's great. But if you don't mind, if we
No more stuff.
need to go a couple minutes over. We might. Jenny, the questions?
Sure. So I know you addressed the different phases for the clinical trial, but it sounds like people are still interested in a little bit more about time frame for implementing these and treating patients.
With COVID, these are hard times, but one of the side benefits is that people are learning a whole lot more about development of new therapeutics following the vaccine trials so closely. Fortunately for COVID, unfortunately for us, the warp speed version of development is really well suited to vaccine development, given the deep understanding of vaccines, how they work in the body, and their safety profile. When we start with a new target, like the ones we described, or a new gene therapy, we need to be prudent about it and establish efficacy and safety in animal models first, and then test it in a small number of patients first. So phase 1 is it safe? And don't want to be treating large numbers of patients before you know it's safe. Phase 2 then is, does it work at the level of, is there enough, developing enough evidence that it could work in order to inspire the longer, larger, and more expensive phase 3 trials, which are designed to convince patients and practitioners and regulators like the FDA that it does work and is safe. So that's sort of the main purpose of the 3 phases. But this does take years. An average drug could be 12 years. Now, the FDA is partnered with academics and industry to find ways to shorten the development process. For particularly severe diseases, this can be shortened. It was a big issue with AIDS a few years ago, and there was definite acceleration seen. And with gene therapies now, particularly gene therapies that affect young children dying, young children in wheelchairs, the whole community, including the FDA, has been engaged in approving early, just because the needs of those patients are so great. And there are forms of genetic DCM that affect children. And we're working closely with pediatric cardiologists and the Children Cardiomyopathy Foundation, similar to the DCM Foundation, to identify what are the biggest needs of infants and children, and in designing gene therapies to address those needs. So the timescale is very different based on a number of factors, but it is years.
Great. We've got a couple more questions, and we actually have sort of hidden the CEO, Faraz Ali, on the webinar, and there's a question. It might be great for him to answer. There's a question. It's in everybody's mind, I think. And you create these gene therapies, small groups, expensive. What's going to happen? How will we be able to afford these therapies? Because right now they're very expensive. Faraz, where do you see this going, and how does the average person afford it?
Yeah, thanks for whoever asked the question among the audience. Hi, everybody. I'm Faraz Ali. I'm the CEO of Tenaya Therapeutics. I've been in the drug development field for 25-ish years, and in the genetic and orphan side of things for a good chunk of that. And this is a conversation about price and affordability that people may feel uncomfortable often asking, and companies may not be uncomfortable talking about it. But I do think it's something we need to be open about and transparent about. And look, let's be clear. It hasn't been good, right? People have had a hard time accessing expensive therapies, whether they were devices like ICDs or drugs. And then there are gene therapies for rare disorders that have been priced in the millions. And I think the you know, it is right now as a company, Tenaya can't really know what the future holds, what you know, how many patients we'll be able to treat, what's going to work, what's not going to work. Our first priority is to advance some really, really good science that can hopefully bring something valuable to patients. And Whit gave you a flavor for some of those things today. Some of those things are going to work, some of the things are not. And probably the majority won't work just based on what we know about how tough science is and how tough clinical development is. Some of the things we're going to work on are going to be for very, very small populations. Phospholamban mutations that Whit mentioned, that's, you know, there are a couple thousand patients in the world, or in the US and Europe. And then some things we're going to work on will hopefully affect millions of patients. What eventual prices end up being here is really in some ways unknowable until we've developed them. We've seen how well they work. Do they really actually change the lives of patients or not? If they don't, they won't get approved, and even if they got approved, nobody's going to pay for them. So it depends on whether it's a gene therapy or a small molecule. Is it a one-time therapy, or is it a chronically administered therapy, that a pill you have to take every day? And it matters whether it's for a small group of people or for a large group of people. So these things are all, you know, not yet known today, and we'll know more as we go along. What I can say, so I can't speak to a specific price for a specific product, but what I can say is that, as Whit shared, our values are centered on the best science for patients. And we intend to keep patients involved along the entire continuum. And that means we'll also hear and understand about insurance. It may seem like a long way away to an approved product, but we will early start to understand insurance and insurance challenges and what difficulties people have had accessing therapies and devices and treatments for, or even heart transplants. By understanding that now, we as a company can be better at anticipating challenges in the future. And hopefully with that information, we may even be able to design better studies that will generate the data that'll support the coverage by the insurance companies, because they're the ultimate gatekeepers, physicians who prescribe and the insurance companies that pay. So in addition to engaging with you, we have to engage with them and understand what will support a prescription from a physician and what will support payment by insurance. And we are already thinking about those kinds of things now. We're already thinking about things like the cost of manufacturing and trying to drive it as low as possible. Because if we do that, then that gives us a better shot in being able to afford to offer future products at lower prices. So all I can say is we're thinking about it. We care about it, because all the science in the world is meaningless if you, as the patients who can benefit from it, can't access it. And as a company, we hope to do better than the companies that have come before us.
Thanks. And we had a few other questions. That was very well put. Thanks, Faraz. You know, we have a couple other questions like that too. So certainly, you know, dealing with, you know, insurance and costs and things are very difficult for chronically ill patients. And certainly, I know myself with DCM. What do we think patients could do? You know, there are issues, as you mentioned. Could we get, you know, again, participating in groups like this, reaching out to you know, your representatives in your state capital or certainly nation's capital, I think is important. But are there a few things as we wrap up here that you guys see about patient advocacy that we as a group and as individuals could do to help affect positive change here?
You know, I would say this talked a lot at the heart meetings about how effective for cancer patient groups have been, so that the whole country really, you know, understands what it means to be diagnosed with cancer, and you know, the life-changing diagnosis that it is, and I think for a variety of reasons, the just the understanding and empathy really for heart disease is not the same. So people hear cancer, there's one emotional reaction, and hear heart disease, doesn't resonate with people as much. And this does have implications, and then that could go right to payers. And we can see that biologic medications, the antibodies, which are also complex manufacturing, expensive to manufacture, but payers are more readily paying for oncology versus cardiovascular type biologics. The understanding of what patients are really going through really makes a difference in everyone's mind as they think about these things. So that's payers, but regulators and scientists too.
Yeah. Thank you. It is a difficult issue, a difficult problem that needs to be addressed, and we will be working on that at the DCM Foundation. There's a couple more questions, but I think we better wrap up. It's 7 minutes past the hour. But again, I'd like to thank Whit. We'll actually answer your questions, those that were not answered. When we send in the next few days the actual recording and the deck, we'll answer those questions. We'll talk with Whit and Faraz and get those answered and send to you with that. The second thing is we'd love thank you, Whit, thank you, Faraz, and for what you do, and thanks to the presentation today. It certainly gives me hope, and it sounds like almost everybody else on the webinar. And we hope to see you soon, and appreciate the time that you took with us today.
Thank you, Greg, and Jenny, for hosting us. We look forward to a long collaboration.
Very good. And thanks for attendees. Have a great afternoon, evening. We hope to see you next month. Bye-bye.